Hi, this is the second lecture on DNA biology, evolution, and immunology. Now, the last lecture we stopped at nucleic acids, and I just want to alert you that we're not continuously just using the same chapter. So if I press these arrows, it's just going to take me to the next section of that chapter. So what you want to do is you want to go up here and look on your lecture study guide, and you can see the next section we're going to do is 3.4. And so I'm going to go down to chapter 3 and section 3.4. And in fact, we're going to 3. Okay, here we go. We can start there. Okay, that'll get you there a little bit faster. So what I wanted to talk a little bit about, I'm going to continue my discussion about DNA and RNA and proteins. And just wanted to touch on the concept of chromosomes. And so the nucleus contains the genetic instructions, the DNA, necessary for the manufacture of proteins. And that DNA is, gosh, one of the videos I gave you said something like, if you took one cell, any one of your cells, take a skin cell, and you unraveled all the DNA in there, you would have a molecule that is 2 meters long. That's not quite, it's like 5 feet long. So one single cell in your body, the DNA in that cell is about 5 feet long. Now there's some amazing chemistry going on that and I guess some of it's physics, that rolls that DNA really tightly and packs it into a microscopic cell. So in addition, and so all that DNA is in the nucleus of the cell. So here we're talking about eukaryotic cells like human cells. So all that DNA is packaged into the nucleus. And in this picture, in the enlargement of the nucleus, they're showing you that you can see this kind of thread-like stuff. That's what we call the chromatin, and that's one form of the DNA. Now the other thing that happens, it's kind of the not quite as tightly bound, folded up DNA. But one of the other things that happens is once cells start to divide, the DNA kind of compacts even more. And it turns out that that 5 feet of DNA in your cell is not just one long strand. It actually is divided into segments called chromosomes. So chromosomes are really just pieces of DNA. And it's just a way to organize the cell rather than have this big, one big long strand getting all tangled up. So you probably remember from another course that humans have 46 chromosomes, 23 you got from your mother, and a matching 23 that you got from your father, your biological parents. And so the chromosomes are just pieces of DNA. Now, if we look at one chromosome, there actually are units of DNA within there that are called genes. Now, a gene is enough DNA to encode for the production of a protein. So here you have a chromosome, and there's many hundreds of genes on that chromosome. And so what it means is that that DNA has enough instructions to make many different proteins. So a gene's a segment within a chromosome or within a piece of DNA that has enough instructions on how to make a single protein. I know I don't have ribosomes highlighted here, but ribosomes are going to come up again, so remind yourself the function of ribosomes. And remember, ribosomes are where proteins are made. I think that's it. So now I'm going to go to, if I look back on the lecture study guide, my next section is 22.2. So I'm going to go back up here and I'm going to find chapter 22 and I'm going to find section 22.2, gene expression. So we're going to build on what we know, what we've been talking about with proteins and DNA and RNA. And so it goes into some detail here. It reviews the structure of proteins. So you can look at that one more time. And I want to kind of jump ahead and talk about gene expression. So gene expression is the process by which you use DNA to make RNA, oh, here's this picture right here. This is gene expression. We use a DNA molecule, and that is transcribed in the nucleus to make a molecule of RNA, and it happens in this example most of the time it's messenger RNA. We're just talking a little bit about that in lab. And that RNA is translated at the ribosomes to make a polypeptide, better known as a protein. So when we talk about the concept of gene expression, we're talking about how DNA is used to make RNA, and RNA is used to make proteins. And so these should look really familiar. Here's our double-stranded DNA. Here's our bases, and they're complementary. A bonds to T, G bonds to C. So what we're talking about when we're talking about gene expression is the process called transcription, which is taking DNA to make RNA, and translation, taking RNA to make proteins. Now this, if you think about these words, we say both DNA and RNA are nucleic acids, and their building blocks are nucleotides. So if I'm going to transcribe something, let's say you're transcribing what I'm saying, and if you're transcribing what I'm saying, you're writing down what I'm saying in the same language. And so when we go from DNA to RNA, the cell's using the same language of nucleotides, and so the word transcription makes sense. When we go from RNA to a protein or a polypeptide chain, we have to change from nucleotides to, remember what these are called? Amino acids. You go from, when you're making RNA, using RNA to make proteins, you go from nucleotides to amino acids. And so we have to translate that into a different kind of chemical language. So the word translation makes sense for that. So when we're talking about gene expression, we're talking about using DNA through transcription to make RNA and through translation to make proteins. And the textbook and lots of resources online go into a lot of details about transcription and translation. But I'm just kind of want you to have the big picture, and frankly, some of the videos that I have in the lab will kind of fill in some of the missing pieces for you, but still keep it at a really basic level. One of the things that you're going to notice here is that when you make, I have this DNA molecule, only one side of the DNA molecule is going to be used to copy the messenger RNA. So in the videos, it's going to show you that this DNA molecule opens up. There's an enzyme called RNA polymerase. It makes a polymer of RNA. And so what you can see is if this strand of DNA is being copied into RNA, if there's a T in the DNA, there's going to be an A in the RNA. If there's a C in the DNA, there's going to be a G in the RNA. So the sequence of the DNA is going to instruct the cell how to transcribe RNA, what your RNA sequence is going to be. The one thing they don't have here is they don't have an A. If there was an A in the DNA down here, there would be a U in the RNA. Remember, RNA can have U's, not T's. Okay, and you'll see more of those details, like I said, in the video. So here is, I wanted to talk about mutations. Now, what a mutation is it's a change in the DNA sequence. A mutation is a change in the DNA sequence, and mutations happen all the time. They, every time one cell divides into two cells, the DNA has to be copied, and that's in a process called DNA replication that we're not talking about here. It's a different process. But every time you copy DNA, there's a possible mistake that's going to happen, and that mistake can result in a wrong base pair being put into the DNA. As a result, you could end up with an RNA sequence that also has a wrong base pair from the original sequence, and that could also result in a wrong um, amino acid in the protein. Now here it's talking about mutations. Mutations means a change in the structure of DNA. And it turns out that not all mutations are bad. I think people automatically say, oh, your DNA changed, you've got a mutation, so that's going to be a bad thing. You're going to get a disease. And because we hear a lot about that. And it's true, there are a lot of diseases that results from mutations. I can think of like cystic fibrosis and Huntington's disease and sickle cell anemia and many, many other diseases that have a genetic basis, and they can be results of mutations. So something went wrong. Some change in the DNA caused a change in the RNA, which caused a change in the protein. And so the protein might not be folded correctly, and it might work incorrectly and have a detrimental effect on the organism. But mutations also give us variety. And so sometimes mutations, they don't seem to do anything. You know, they just kind of are different. So if you have you know, we have a lot of variety in hair texture. We have fine hair and thick hair and straight hair and curly hair. And all of those changes in hair variety are a result of mutations that occurred in some organism a long time ago, and they were passed along. And so most of these mutations seem to have little or no effect on the health of and the ability to reproduce for the individual. So not all mutations are bad. Some just result in variety. The other thing is some mutations are actually good because they allow for some characteristic that gives a species an advantage. And so, you know, we talk about probably one of the more visual ones is we talk about like giraffes that mostly eat food in trees. So a tall giraffe is going to get more food and probably be stronger and reproduce more. So having mutations that result in taller giraffes can actually benefit the population and help the taller ones reproduce. So no, mutations, even though it sounds like a bad thing, it might have a detrimental effect. It might have no effect on the resulting organism, or it might even have a beneficial effect that allows the organism to survive and reproduce more effectively.